Good afternoon, everyone. It is a pleasure and a privilege to introduce today's seminar speaker, Dr. Toshiko Tanaka, though many of us know her simply as Tosh. It is a special moment for me, as Tosh was once my student here at the HNRC. We were remembering how long is the last time that we saw each other in person, and it happens to be too many years. Anyway, after she left the HNRC, she changed the flavor of her research, and I say flavor because her thesis was on taste. And she, after graduating, she took her passion for understanding the biological underpinning of aging to the NIA. As a postdoctoral fellow, she worked with Dr. Luigi Ferrucci, where she deepened her expertise in the fascinating intersection of nutrition, molecular epidemiology, and aging. Now, as a staff scientist in the Longitudinal Study section of the Translational Gerontology branch of the NIH's Intramural Research Program, so this is almost as long as our HNRCA at Tufts, Tosh continues to advance our understanding on aging through a holistic, integrated approach. Her work and her commitment to the field have become valuable contributions to both science and the well-being of aging populations. Please join me in welcoming Tosh.
Thank you. Yes. Thank you, Jose. Thank you for that kind introduction, and thank you for the organizers for inviting me to give this talk today. I think the last time I spoke at Tufts was over 10 years ago, and my graduation was even longer than that. So it's a pleasure to be back here and to share with you some of our work that we've been working on, where we implement concepts of geroscience and epidemiology, and we are in the business of identifying aging biomarkers in human studies. So I've structured this lecture in 3 main sections. First, I'll be talking about aging and geroscience and giving an example with mitochondrial function. And then I will start to describe some of our studies where we use proteomic data to identify biomarkers of aging, specifically focusing on studies that looked at chronological age as a phenotype and also frailty. And at the end, I'd like to discuss where we're going and what we are trying to do in our group to identify and refine biomarkers of aging using longitudinal data. But I wanted to take a moment here to share this slide. This slide actually was shared by Dr. Corella here, who came to the NIH to give a talk, and she showed this picture, and my supervisor, Dr. Frucci, took a snapshot of it and sent it to me. This is the Nutrition and Genomics Lab at the HNRC. I don't know what year it was, but it's in the early 2000s, I'm sure. And I just wanted to impress upon the students who are at Tufts right now that you're in a really, really good place. This is really where my career began. And, you know, really nurture the relationships that you make at Tufts, because a lot of my classmates have become my collaborator, and also many of my mentors have become, continue to be my mentor, but also have become collaborators. So especially if you stay in nutrition research, you're going to have a lot of interaction with other jumbos. And so please, yes, recognize that you're in a very, very good and special place. Okay, so let's talk about geroscience. I don't think I need to talk about why aging is an important feature to this group, but here is a study in the UK Biobank that shows that age, advanced age, is one of the major risk factors and a consistent risk factor for many chronic conditions. Here is showing the incidence of different major chronic diseases with age, and you can see that there is a linear and almost, in some cases, an exponential rise in the incidence of different diseases. This black line here is a line, or the brown line, sorry, is a line for health span, meaning the occurrence of any of the diseases that you find in this figure. And you can see that with advanced age, you have higher incidence. However, if you're someone in epidemiology and interested in a specific disease, such as diabetes or stroke, often in our models, we adjust out the effective age by including age in the model. The idea of geroscience is to actually focus on age as a means to understand why these diseases increase with age. So, with the understanding that there is this basic biology that underlines aging, and by uncovering the aging mechanism, we can begin to understand a common thread that is shared between different chronic diseases. So, to that end, in terms of biology of aging, there's been several review papers. This one is from Dr. Oten, where he describes 9 hallmarks of aging, and it's become kind of a famous paper in the aging field. Um, each of these 9 different pathways have been shown both in animal studies and to a certain extent in human studies to be associated with aging. And our goal is to be able to measure these processes in humans. What's important to understand is that these aging mechanisms have a hierarchical relationship with what we call the metrics of aging. So, at the root, what we call biological aging is this equivalent to the hallmarks of aging, which are the mechanisms that are related to aging. And then with an accumulation of damage that occurs in aging, we start to see an effect in phenotypic aging. So I think about these as intermediary biomarkers of aging. So things like body composition, cardiac output, or muscle quality, and things of that nature. And finally, we have functional aging. These are the things that we want to delay or prevent. So we have cognitive impairment, frailty, chronic diseases, and things like that. Another important concept is that these different metrics of aging have a temporal relationship. So here again, we have the 3 layers of aging. And at the root, we have the biological aging, which are the mechanisms that probably begin to decline earliest. But at the earlier time frame, there's enough resilience in the system that we see no difference in phenotypic or functional aging. But as damage accumulates, you begin to see at a threshold a decline in phenotypic aging. And as accumulation increases here at a critical point, you start to see effect in functional aging. So currently in clinical medicine, where we intervene is here. When we begin to see changes in things we can measure. So you might begin to see increase in blood pressure or high glucose or dyslipidemia, and then you are put on medication and things like that. But at this point, you can see that the accumulation of damage that happens into the biological aging has already occurred. So it might be more difficult to reverse the damages when there's a lot of accumulation that already happened. And so by being able to measure biological aging, perhaps we can intervene earlier in the spectrum so that we can prevent damage before it actually occurs. So what does a measure of biological aging look like? We understand that aging is heterogeneous, so people experience aging in different ways. So here we have three individuals. These are not the same people, but I'm trying to show what we're trying to express here. So if we look at chronological age, we could take a snapshot of these three individuals at the same time, their same chronological age, earlier in life at 25 and midlife at 50, and then later in life at 75. So these numbers don't inform us too much about the health of the individual. So if we had a measure of biological age, we might be able to say if one person is aging faster or slower than their actual age. So for example, the person in the top, you can see at age 25, their biological age is 20, so a little bit younger than their actual age. And you see this trend persist with the passage of time. But I think the most important is this person at the bottom. So at age 25, where the individual is seemingly healthy, you see that already their biological age is a little bit older. And then in midlife, you see a greater change or difference between their chronological age and biological age. And you see that by 75, you see this huge difference in their accumulation of damage. And so, if we were to have this measure of biological age, we might look at this person and start to think about what is going on in terms of their rates of aging. So here, just to define, chronological age reflects the passage of time, whereas biological age is a measure of damage accumulation throughout the lifespan. And so the goal of our group is to be able to measure and quantify this measure of biological age. So going back to the hallmarks of aging or the aging mechanism, one approach that we have taken is to try to, for each of these categories, come up with a measure that could be applied in the epidemiological study. Our group is very interested in mitochondrial dysfunction or mitochondrial function. So I want to take a moment to talk about what we've done with mitochondrial function in humans. So the mitochondria is considered the powerhouse of the cell. So it's a very important in producing energy. So mitochondria is found in higher abundance in tissues with high energy demand. So things like muscle, but also the heart and liver. And we know that mitochondria or the ATP is very important in aging because we see that traits that are related to muscle have important implications or changes with age. This is an example with grip strength. This is a measure that we do commonly in aging studies because of the ease of measurement. We have this dynamometer that can measure grip strength, and you can take that to the field quite easily. So this graph is results from a meta-analysis of 11 studies in the UK by age, which show that the peak of muscle strength measured with grip strength is 31 in males and between 30 and 40 for females. And you see this gradual decline with age, both in men and women. And there are important health implications of decline in muscle strength and also muscle function. So this is another important aging physical function test. Gait speed, this is also a commonly measured trait in a lot of aging studies. So we ask participants to walk a certain distance. We have like a speed up period where people can get going and then they'll walk for 10 meters or 5 meters, and then we can measure their speed at which they walk. And what's interesting, and actually when I first learned about this, I was very surprised, is that the walking speed in midlife, so this is starting at 65, but this is also true for earlier in their lifespan, is very correlated with mortality rates. So this is looking at men and women and looking at gait speed at different stages and looking at their median survival. And you can see this dose-dependent association with gait speed where faster walkers have decreased risk of mortality compared to those who are walking slower. So these phenotypic association point to the fact that mitochondria may be an important factor in aging. So we've implemented a technology that allows us to measure mitochondrial function in vivo. In order to do this, we need to understand the phosphocreatinine shuttle. So when the muscle contracts, the ATP that's used by the sarcolemma is not directly from the ATP from the mitochondria, but it comes from the phosphocreatinine. So when ATP is needed by muscle contraction, the ADP is combined with the phosphorus from the phosphocreatinine by the creatinine kinase to produce the ATP. So as you have muscle contraction, you have depletion of phosphocreatinine and increase of creatinine. And in order to recover phosphocreatinine is dependent on muscle or mitochondrial function. So through oxidative phosphorylation, you have a production of ATP, and this ATP becomes the donor of the phosphorus to recover phosphocreatinine through the mitochondria kinase. And so by measuring phosphocreatinine, we can indirectly measure the mitochondrial function or the rate of oxidative phosphorylation. So again, here is the pathway that I just talked about. So what we do is we have participants that are put in an MRI, and there's a coil that measures the level of phosphocreatinine in the thigh muscle. And once they're in the MRI, they are asked to exercise. So it's kicking, they're asked to kick. And while they are exercising, you can see the level of phosphocreatinine deplete. So here, this reaction is happening as contraction occurs. So you can track this real time. And when you reach a 60 to 70% depletion, you ask the participants to stop. And once they stop, you have this process of recovery of phosphocreatinine, and this rate is proportional to the oxidative capacity of the mitochondria. So using this methods in our study, we were able to show that this on the y-axis, we have kPCR, which is a measure of oxidative phosphorylation. In the mitochondria, higher values is quicker recovery and better mitochondrial function. And in both men and women, we see that there is this age-associated decline in mitochondrial function. We also have linked this measure with functional measures. So we find that mitochondrial function is associated with walking speed. So in the same individual at the same visit, we have a measure of pace of walking. We have different tests that measures usual walking pace, but also fast walking pace. So sometimes when someone is highly functional, by stressing the system and asking them to walk faster than their usual pace, we can see a decline, a little bit more sensitivity. And this association between mitochondrial function and walking speed was mediated by muscle strength here. So it seems that mitochondria has an effect on function and also muscle strength. The issue with measuring mitochondrial function is that you need a lot of equipment. You need an MRI, and it's very, very difficult. So we are, in addition to collecting more data on mitochondria, we look for alternative biomarkers of aging. And I have been involved in leveraging omics data to identify aging biomarkers. So a couple of weeks ago, I believe that Dr. Talagakar, who's also a Tufts alumni, had presented some of our work on metabolites and aging phenotypes. So today I'll be focusing on proteins. And proteins is a very interesting omics layer. I think it's a data that's very nice to work with. It gives you a lot of information about the pathways that are involved with aging. And the interpretation is quite interesting because you're looking at the actual molecules that mediate the pathways. Also, from a clinical standpoint, proteins are very commonly used as biomarkers for different diseases. So if we think about translation, it's proteins and metabolites may be an easy way to implement in the clinic. So here we're looking at proteomics, and one of the challenges of proteomics is that there is this dynamic range of proteins in the plasma, which makes it challenging to measure proteins. What this means is that, for example, albumin, which is the most abundant protein in the blood, takes about 57% of proteins that is circulating. And then you have about 10 additional proteins that contribute to 90% of proteins that are circulating. But really, what we want to measure is the remaining less than 10%. But when you have these proteins that are really abundant, they confuse the signal, and it makes it hard to measure these lower abundant proteins and the proteins that you might be interested in. So these include many of the cytokines that are important, inflammatory markers that are important in aging. What has really helped with the progress of proteomic studies in aging and in other fields is the advent of targeted technology. I'm just showing an example here of SomaLogic, which is the data I'll be sharing today. This is an aptamer-based technology where you can measure a large number of proteins in plasma. Here is just to show how there's been a linear increase in the number of proteins that we are able to measure in the blood. We initially started with the version 3 or the 1.3K, which measures 1,300 proteins, which we thought was a lot. But the most recent version includes 11K, which is over 10,000 proteins. So this is a lot of proteins, and you really don't need a large volume of blood. And so the ease of measurement has really accelerated discovery. So it's really exciting to be in this field at the moment. Okay, so the project that I will show you are these two papers that were published in Aging Cell and eLife. So before I talk about the studies, I just want to orient you on the two studies that we've used for these projects. One is the Baltimore Longitudinal Study on Aging, or the BLSA. The main aim of this study is to understand aging. We characterize both physical and cognitive changes that occur with age. This study began in 1958, and BLSA is one of the longest studies of aging in the United States. The study is based in Baltimore, but here you can see the figure of where all our participants are. Many of them, again, are in the Baltimore, Virginia, DC area, but you can see we have a pretty big cohort in Florida and Canada, so not Canada, California, I'm sorry. So perhaps some of these participants have been in the studies for 20, 30 years, so there is some migration. It is a rolling enrollment study, which means each participant is on their own schedule. So at enrollment, we look for people who are over 20 years and older. and in general good health. It's actually a pretty strict criteria. They must be free of most chronic diseases except for controlled hypertension, no cognitive impairment, no physical functional impairment. And then we follow them at different time frames depending on their age. If they're under 60 years old, we follow them every 4 years. If they're between 60 to 80 years old, we follow them every 2 years. And if they're older than 80 years, we follow them every year. And at the study visit, the participants come into our clinic for 3 days. So we do extensive assessment of both physical, functional, and cognitive functions, and we collect many, many biosamples. The second study is the Inchianti study. The aim of this study was to understand the risk factors that influence walking ability in older participants. This is a study that is done in Tuscany in Italy. The participants are recruited from 2 towns and cities in the Florence area. So we have one where it's more urban and another that is more rural or suburban. And 50% of the population study sample come from the city and 50% come from the rural setting. The study itself has focused on participants who are over 65, but there are some people who they sampled younger people, but the population or the sample size is smaller. The study design is a baseline study happened between 1998 and 2000. There was 1,453 individuals that were recruited, and they are also followed periodically. The first 3 follow-ups were between 2 and 3 years apart, and then there was a little bit of a gap, and we have follow-up 4 and 5. The last one was in 2018. However, we do follow the participants to get some endpoints such as mortality and dementia. So we do have some data beyond 2018. So using these 2 studies, our aim was to understand the proteomic profile of age using the targeted proteomics platform. So we have studied samples from BLSA and Gestalt. Gestalt is another study, it's a sister study of BLSA. The protocol is very similar, and the sample participants are very similar to the BLSA. They are followed every 2 years rather than every the staggered follow-up time. But using these 2 data sets, we identified 240 healthy participants. So participants who were free of functional and cognitive impairment. The age ranged from 22 to 93 years of age. And then we had the SomaScan 1.3K. And the Inchianti study, we used baseline data. We had proteomic data on 997 participants in the Inchianti study. And again, we had the same SomaScan platform of 1.3K. So the main aim of these studies were to identify plasma proteins that are associated with chronological age. And the second aim was to create a multi-protein index of chronological age, or what we call the proteomic clock. The results from the first analysis, the first study we did was in the BLSA/Gestalt study. We found 217 proteins that were associated with age. So in this volcano plot, these individual dots are the regression analysis between the protein abundance and age. So all the dots to your right are proteins that are found in higher abundance with older age, and proteins to the left are proteins that are found in lower abundance with older age, and the minus log 10 to the p-value indicate the significance of the association. So the most significant protein is GDF15. This is a protein now that has been replicated in a large number of studies. We followed a similar analysis in the Inchianti study, and you can see many of the proteins that are significant are similar And when we actually look at the data, of the 217 proteins that were associated with age in the BLSA and Gestalt, 211 or 97% are replicated in the Chianti in the consistent direction. And conversely, when we look at the proteins that are associated in the Chianti study, 330 or 80% are replicated in the BLSA. So we have here two populations that are geographically quite different and also very different from a health standpoint. And what we see is very consistent proteomic profile. We did a enrichment analysis of the age-associated proteins, and we find several interesting pathways that are enriched based on the proteins that are age-associated, including measures of inflammation and axon guidance, so cognitive function. So this will come up a little bit later on in the talk. The next thing we did is generate a multivariate machine learning model, or what the field now is calling clocks. Here, what we do is instead of looking at individual proteins, we look at multiple proteins. So using a learning model, so an elastic net or lasso, we identify multiple proteins that can predict your target trait, in our case, chronological age. So here we have the measured chronological age and the predicted age that is based on your data, so proteomic data. And then if we have this regression line, for those people whose predicted age based on proteomic data would be considered as aging faster, and those who are on the lower end or predicted to be younger than their actual age would be considered to be aging slowly. So we tested this hypothesis in the BLSA and Gestalt, and we created a 76 protein clock using elastic net regression, and we were able to create a index that was highly correlated with age. And when we applied this to the Chianti, we saw that the algorithm that was built in a healthy population was also associated in a more general population. And what was interesting here is that when we focus just on the healthy population or individuals that have no comorbidities, the correlation was higher at 0.91 compared to those with comorbidities, where the correlation was 0.76. Also, we noticed that those with comorbidities, their predicted age was older. So this idea of deviation being higher in those with comorbidities. So we used the clock concept and looked at the deviation between the predicted and the actual age and looked at future outcomes. So here we focused on all-cause mortality and multimorbidity. So for all-cause mortality, for those who were under 65, we saw no difference in the fast agers and the slower agers. But in those who were over 65, those who had higher aging rates based on the proteomic clock, the risk of mortality was higher. Similarly, for multimorbidity, those who were predicted to be older had a faster accumulation of diseases. So these data suggest that this deviation does include some information about health. And this has been replicated in other studies. This is just an example from Tony Wyeth-Corey's group. This is a clock that was developed using a little larger proteomic platform. So this is the 7K SomaScan, and they did a very similar process where they took the 5,000 proteins and created a proteomic index. Again, the Pearson correlation is very, very high. When they looked at the deviation, they found that it was associated with cognitive function and physical function. So it seems that this clock concept does work in terms of association with important aging phenotypes. I just wanted to show here this new well, somewhat new data from Tony's group. This was a very beautiful paper. It's the organ specific clock that developed. They leveraged data on gene expression, GTEx, and they categorized the different proteins that's measured by the SomaScan into different organ specific proteins. So if the expression was higher in one organ and not in the other, that protein would be assigned to that specific organ. And what they find here is quite interesting. First is that the organ specific clocks were not correlated. This suggests that different organs age at different rates. Another interesting thing is that 23 of the population were extreme organ agers. So not too many people are, are aging too quickly. And another thing that was interesting is the prediction of outcome was better for the clock that was developed in a relative or relevant organ. And what I mean by that is this figure here. Here is comparing the different organ clocks with congestive heart failure. And the most significant predictor was this heart clock here compared to the others. But if you look at a more general outcome like all-cause mortality, this conventional clock or organismal clock was the most significant. So this concept has been really was very unique and very clever, I have to say. And it has led to development of other organ clocks. And it's beginning to become clear that these organ clocks are predictive of specific outcomes. That makes you think, what are the things that affect the organ clock? So I want to share with you some preliminary data that we have looking at the association of diet and these proteomic organ clocks. Going back again to the BLSA and the Inchianti study, we used the proteomic data. In the BLSA, since the original paper, we now have data using the SomaScan 7K. And then we're also using the Inchianti 1.3K. We have dietary assessment in both studies using a food frequency questionnaire. From Tony's paper, we are able to pull out the different proteins that are related to the organs. The organs with the asterisks are the ones where the model includes sex. So here, I just wanted to show that the BLSA had all the proteins that were in the original paper. The Inchianti, because it's a 1.3K, has fewer proteins. But the correlation was surprisingly very good, even with these fewer proteins. We looked at two dietary patterns, the Mediterranean like diet scores and the MIND diet. So these two scores are similar. The difference is that the MIND score emphasizes food items that have been associated with cognitive function. So the correlation between these two, there is a correlation about 0.45. However, these again emphasize individual food items. And we found something very interesting here. When we look at the association of adherence to the Mediterranean diet score and the MIND diet score, we see that for BLSA, the most significant association was found in the liver. The way we look at this is that with higher adherence, there's a negative effect. So the liver, with better adherence to the Mediterranean diet, the liver is aging slower. And we also see this in the Inchianti study for the MDS. For the mind, we see this huge effect for the liver, but the mind was not significant. The interesting thing here is that when we look at the brain clock for MDS, there was no significant association. However, for both the BLSA and Chianti, we see that adherence to this MIND diet score was associated with slower brain aging. So we thought this was quite interesting and we're pursuing this a little bit further. So So hopefully you'll be seeing this data pretty soon. Okay, so I want to switch from chronological age to another important phenotype of aging, and which is frailty. So frailty is a state of increased vulnerability to stressors, and it's an important geriatric syndrome. So those who are on the higher spectrum of frailty have worse aging outcomes, such as falls and hospitalization. So we want to understand ways to prevent frailty, but also to quantify it better. There are different definitions of frailty. In the BLSA and in the Chianti, we've used the deficit accumulation model where we add up the number of deficits. In our frailty index model, we include measures of chronic disease, ADL, IADL, self-rated health, cognitive function and depressive symptoms, unintentional weight loss, sedentary behavior, physical function, assessed by walking speed and grip strength. So for every individual, we count the number of deficits. For example, if you have cancer, you would have you would have one score added to your frailty index. Or if you're unable to walk up 10 flights of stairs, you would get an additional score added to your index. So we count the total number of deficits and express this as a ratio divided by the total number of deficits. So if you have a score closer to 0, you would be robust, and a score closer to 1, you would be frail. Using this concept, we looked at the Chianti study to identify proteomic profiles of frailty index. So in this study, we have a 42-item frailty index calculated at baseline and in the 3 follow-up periods. We have plasma proteomics assessed at baseline. Again, this is the 1.3K. And we use linear mixed effects model to identify proteins that are associated with trajectory of frailty index. And we find that there are 92 proteins that were associated with frailty index. Here again, these are the proteins that are associated with faster accumulation of deficits. And these are the proteins that are associated with slower accumulation of deficits. Here is just to show what the trajectories look like for the most significant risk protein and most significant protective protein. We also did an enrichment analysis of the proteins that are associated with frailty index, and this is what we found. We found several familiar looking pathways in addition to novel pathways. This is just as a reminder, this is from the chronological age analysis where we did an enrichment analysis. As you can see here, we have complement coagulation cascade in common and also the cytokine receptor interaction in common. However, here we have a few novel pathways being highlighted using frailty index. So I think what this highlights is that it's really important to target different aging traits in order to get a catalog, a comprehensive catalog of potential biomarkers of aging. Okay, so we're in our last phase of the talk. I want to talk about what we are thinking in terms of trying to refine phenotypes of aging. And really what we want to do is to leverage the longitudinal data that is available to us in the Baltimore Longitudinal Study on Aging. This is work done by a postdoc that was in our group, Dr. Kuhl. Here, he took the BLSA and thought about it in 4 domains. And for each of these domains, he investigated the age-associated trajectories and looked at the trajectories by sex and by age. And you can see that there are different shapes in terms of how these traits change within each domain. And by creating, for each traits, he looked at the change by sex and by age and categorized people if they were in if the rate of change was higher than the average of someone who is the same sex and age or lower. So those people on top are the ones that are aging faster for that specific trait. And the ones at the bottom are the ones that the trait is changing slower than those who are the same age and same sex. And for each domain, he did this for all of the traits that were available to him. And one thing he found is that the rate of change that is a composite score for each of the domains were not correlated with each other. So this is reminiscent of what was found with the organ clocks, where each of the organ clocks were not correlated with each other in Tony's paper. And we find the same when we look at phenotypes by these different domains. So he created this global longitudinal phenotypic score by collapsing these four information into one score. And what was interesting here is that he found that this longitudinal phenotypic score was predictive of cognitive function, physical function, and multimorbidity index. So here it is showing that an increase in this an increase in 1 point of this global longitudinal phenotype score is equivalent to an increase in 11.43 chronological years. So you can see the effect size in chronological years for these different traits. And when he compared this to a score that was based on cross-sectional phenotypes, he found that the longitudinal phenotype score was more predictive. And you can see the estimates in the cross-sectional phenotype score compared to the longitudinal phenotype score. He also compared this phenotype score with some of the more famous clock. This is an epigenetic clock, which is based on CPG sites on the DNA, and he found that this longitudinal phenotype score performed better than Grim Age, which is one of the epigenetic clocks here. So we think that by leveraging longitudinal phenotypes, we can create a better trait that tracks or is able to capture biological aging. So in summary, I hope I was able to express to you that mitochondrial function can be measured in vivo and it declines with age. We've shown that this mitochondrial function is associated with important aging outcomes. However, because mitochondrial function, the way we measure it, can be quite cumbersome, we're hoping to be able to capture this in another way, perhaps using omics. We also observed that there is a consistent proteomic profile of age in different studies that have different characteristics. The plasma proteins that are associated with age are quite consistent. And we have found this within our studies, but also across different studies that have used the SomaScan, but also other targeted proteomic platforms such as Olink. This proteomic clock concept, where we try to predict our trait using omics. The deviation or the clock part has some information about biology. We've been able to associate it with mortality and multimorbidity. Other groups have shown association with disease outcomes. And so there seems to be utility of these clocks. And there's always new papers coming up with newer clocks with better algorithms. So it's in a very exciting field to be in right now. And finally, I think it's important to be able to target different aging phenotypes. I like frailty index because I feel that it is a global assessment of how we age, and other people have used other traits, maybe focusing on domains or organs that they're interested in. Finally, I think there's a computational challenge in trying to implement longitudinal phenotypes. There are now increasing data sources with longitudinal phenotypes. So there's challenges in unifying the different cohort studies so that we can have larger sample sizes, but also places to replicate what we find in one study. So I think that if you're a new investigator studying computation, in order to leverage all the rich resources out there can be beneficial for your career moving forward. So with that, I'd like to acknowledge our group, my supervisor, Dr. Luigi Ferrucci, and a co-director of BLSA, Dr. Simonsek, and my co-workers here at the NIA, as well as core facility. We actually have the SomaScan center here at the NIA. So we're doing a lot more phenotyping. Also, I'd like to acknowledge the participants of the studies that we use. Here is where I am now. This is where BLSA participants come in Baltimore. So if you ever have a chance to come to Baltimore, please come visit us. Thank you.
Well, Tosh, thanks to you. It has been a really outstanding lecture. And now I forgot to say at the beginning that those with questions, they can post them in the Q&A. And we have a few. The first one is from George Mason. Do any of the cohorts in which clocks have been developed possess multi-generational data? If so, has anyone calculated the contribution that inherited features are determinants of proteomic aging?
So we don't, I'm not sure if there's been genetic studies of proteomic aging, but I do know there's been several GWASs. I know this is not generational. It's not a family-based genetic study, but there have been GWASs of the epigenetic clocks. And I know that for Grimm age, I think the most significant was a gene that had to do with telomeres, I believe. But yes, there's been interest in identifying genetic correlations with these clocks.
Thank you. And now from Christopher Wiley. Hi, Tosh, lovely talk. I agree with him. I love the idea that a protein clock can be used to predict biological age. Do you think that this approach offers some superiority over methylation clocks in that it might be easier to identify functional biomolecules that could be targeted for intervention?
Yeah, honestly, I think that all the omics give you slightly different information about the biology of aging. I agree with you in that I love working with proteomics data because it's easy to interpret. When you have the name of the protein, it immediately gives you some ideas of the pathways that are involved. Whereas when you're looking at methylation, you have to think about, which gene is it affecting and things to that nature. In terms of information, when we look at the correlation between the proteomic clocks and the methylation clocks, sometimes there is correlation, but sometimes there is not. And so that says to me that these different clocks are measuring slightly different things. It's hard to say which is better over the other. I think that there's going to be the next generation of clocks that are coming probably will use multi-omics. And so we'll see. I imagine there's going to be many, many clocks that are, if we were to translate this, I think that we will be measuring many, many clocks rather than just one clock. It's not going to be one clock that rules them all. It will be a lot of them.
Thank you. And from Sai Dash, thanks for the interesting talk. The OXPHOS data that you show declines with age also showed great variability, with many in the 60 plus still having excellent functioning of this process. Has this variability been further examined?
Yeah, we are actively looking at this. I think the BLSA is a very unique study in that um, the older people are healthier actually than our younger people, especially the ones that are coming into the study that are, as older individuals. So if they started the study at say like 30, it's easier to meet the inclusion criteria. But if they're coming into the study at, let's say, in their even their 50s, 60s, or 70s, and they are able to meet the criteria, that means that they're kind of a special kind of person. They probably, a lot of them have high levels of physical activity. But yes, we are definitely trying to decipher what are the factors that contribute to the variability in mitochondrial function, and how some people are able to maintain their level of oxidative phosphorylation even at older ages. But I think physical activity is the key.
Michael Mount, has MRI been used to measure the mitochondrial function of the eye or brain, as done with the muscle?
I don't know about the eye, but I know that there is great interest in measuring mitochondrial function in the brain, and that's an active area of research in trying to get mitochondrial function in brain using MRI. But I don't know. I don't know too much about it.
Okay, and again from side, from the proteomic profiles of aging, are there subsets that are more responsive / promising to change and therefore interventions?
Mhm. So again, I think there's a little bit more information with the epigenetic clocks. I know that there's a paper out there from Dan Belsky, I think, that looked at the epigenetic clocks in the calorie study. And I believe the Dunedin clock was most responsive. I think that that's the question we need to answer. There's a lot of intervention studies that have biorepositories. So hopefully they are investing, well, I know that they're investing in measuring many of these omics so that we can identify which ones are the most informative in terms of understanding when someone is more responsive or not. So that's something that is, I think, an active. We'll be hearing a lot about it in the next few years, I think.
Thank you. We are approaching, oh, excuse me. I was going to ask you something, but Sarah Booth has another question. Great talk. Mood and depressive symptoms appear to be an emerging area in measures of aging. Are there measures currently used in these longitudinal studies that, in your opinion, robust and should be included in studies moving forward?
I'm not as familiar with depressive symptom. We have the CESD. Our population is robust from very many different um, domains, including cognitive or psychosocial type of domains. So they perform quite well in terms of depressive and mood symptoms. I think that CESD might be a good measure to have, just because I know a lot of studies have it. So if you needed to do replication, you have the ability to do so, but I'm not as familiar with any of the other tests.
Thank you. And a very short question from me.
Mhm.
I am curious about the sex as a variable, because the data that you have presented I think all of them are males and females combined.
Mhm.
Have you looked at differences
Yes,
between
so
sex?
in that first, in the papers that I showed you, we did look at interaction between age and sex, and there are a handful of proteins, sex hormones and things like that, that do differ by sex. I have not analyzed specifically stratified analysis. I think that's something that should be done, and these scores, there might be organ specific scores and also maybe sex specific scores or even age specific scores, right? And so I think as we move the field forward, and actually there's a huge push to really harmonize across the different data sets. I think we'll start to see more of these um, targeted type of clocks. Yeah.
Okay. Well, the time has arrived that we have to say bye. Again, thank you very much. It has been truly great. And hopefully, we'll see you in Boston next year.
Yes. Thank you so
Thank
much, everybody.
thank you.
Yeah. Bye.